So moving on to section 5, we're going to talk about lipid metabolism. Lipids can provide a greater amount of energy than breakdown or catabolism of glucose or protein. So we can get about 9 kilocalories per gram of fat, where we only get about 4 kilocalories per gram of carbohydrate or protein. Most of the products of fat digestion are transported in those chylomicrons that we talked about in the digestive system chapter. And then eventually we're going to have enzymes that hydrolyze those lipids, giving us fatty acids and glycerol that can be taken up and utilized by our body cells. Only our triglycerides are routinely used and oxidized for energy. And we have two different pathways because they have two different building blocks. So we have a pathway for our glycerol and we have a pathway for our fatty acid chains. So when we're talking about the glycerol breakdown, glycerol is going to be broken down into glyceraldehyde 3-phosphate. This is really important because glyceraldehyde 3-phosphate is a glycolysis intermediate. So eventually this can be converted into acetyl-CoA, which can go into the citric acid cycle. We only get about half the amount of ATP as a product because we only have half of a glucose molecule. We only have one acetyl-CoA entering into the citric acid cycle, where we had 2 acetyl-CoAs that were able to enter in the citric acid cycle from glycolysis from a glucose breakdown. The second pathway is going to be our fatty acid breakdown. And fatty acids undergo beta oxidation. So we're going to get our fatty acid chains that are broken into 2 carbon acetic acid fragments. We have our coenzymes FAD and NAD plus that are going to be reduced. And each acetic acid molecule is going to fuse with coenzyme A, and we're able to form acetyl-CoA from the fatty acids as well. When we're talking about beta oxidation, that's only referencing the fact that the carbon in the third position, which we call the beta position, is oxidized each time a 2-carbon fragment is broken off. But regardless, we're able to produce acetyl-CoA that can go into the citric acid cycle. So again, kind of get an idea of what's going on, nothing specific. We have to go a different pathway for the glycerol component of a triglyceride than we do for the fatty acid components of a triglyceride, but ultimately we're able to produce acetyl-CoA from both of these components, which can then go into the citric acid cycle. So this is showing you the triglyceride breakdown into glycerol and the fatty acid chains. The glycerol becomes a glycolysis intermediate that can go through glycolysis to produce pyruvic acid. But we only are able to produce 1 pyruvic acid, so we only get 1 acetyl-CoA that can go into the citric acid cycle. Our fatty acids, you don't need to know the specific products of their breakdown. Just understand that they're eventually going to be converted into acetyl-CoA by a process called beta oxidation and then able to enter into the citric acid cycle. So just some terminologies. We've already talked about beta oxidation. That's our ability to take those fatty acid chains from the triglyceride and turn it into acetyl-CoA that can go into the citric acid cycle. We also have lipolysis, and we're talking about the breakdown of lipids to fatty acids and to glycerol. And then we have lipogenesis, where we're actually going to form lipids from the acetyl-CoA and glyceraldehyde 3-phosphate. So let's start with lipogenesis. So this is also referred to as triglyceride synthesis. We would only go through lipogenesis if we already had high levels of ATP and high levels of glucose. Because we'd want to form triglycerides, we wouldn't want to break them down. So high acetyl-CoA and glyceraldehyde 3-phosphate is going to go through the triglyceride synthesis pathway. Dietary glycerol fatty acids that aren't needed for energy are going to be stored as triglycerides. So about 50% is going to end up in our subcutaneous tissue, and then we're going to kind of make these fatty deposits in various other parts of our body. So 50% ends up in our subcutaneous tissue, the other 50% will go elsewhere in the body. Glucose is easily converted to fat because acetyl-CoA is an intermediate in glucose catabolism. And remember the starting point for fatty acid synthesis. When we're talking about lipolysis, we are talking about the breakdown of stored fats into the glycerol and fatty acids. So basically, this is the reverse process of lipogenesis. The fatty acids and the glycerol are going to be released into the blood to make sure that we have enough fuel, fat fuel for aerobic respiration. Lipolysis is going to be accelerated when our carbohydrate intake is really low. If we're talking about fuel preference, our liver, cardiac muscle, and resting skeletal muscle actually prefer fatty acids as a fuel source. So we have beta oxidation of the fatty acids, which we talked about previously. This produces huge amounts of acetyl-CoA, which then can enter into the citric acid cycle, only if we have enough intermediates. So remember, we needed oxaloacetic acid to combine with acetyl-CoA in order to make citric acid. So if we don't have enough oxaloacetic acid available, then we can't go through the citric acid cycle regardless of how much acetyl-CoA we can generate. So if we don't have enough oxaloacetic acid, our acetyl-CoA will accumulate. And if it accumulates, it's going to be converted by a process called ketogenesis in our liver into ketone bodies, also called ketones, that are going to be released into the blood. Clinically speaking, when we have ketone bodies that accumulate in the blood, we get something called ketosis, which means we're going to have large amounts of ketone bodies that are going to be excreted into the urine. Typically, you're going to have ketosis if you're starving or have diabetes mellitus, and you can also have this if you are restricting the amount of carbohydrates you're taking into your diet. Most ketone bodies are acids, so ketosis can also lead to metabolic acidosis. And what happens if we get into metabolic acidosis is that our body's buffer systems that we've talked about previously, they can't tie up those acids or those ketones fast enough, and your blood pH is going to drop, and sometimes it can drop to dangerously low levels. So this is the pathway. Again, you don't need to know the specifics, just kind of understand that we have two pathways for the components that make up triglycerides, glycerol, and fatty acids. Fatty acids go through the process of beta oxidation to become acetyl-CoA. Glycerol can be converted into a glycolysis intermediate in order to produce pyruvic acid, which is eventually turned into acetyl-CoA. So synthesis of structural materials. So all body cells are going to use phospholipids because they're components of the plasma membrane. They also need cholesterol, which is considered a lipid, as a component of plasma membranes. We already talked about phospholipids being really important in the myelin sheaths and neurons back in AMP1. We also need these for other organs like our ovaries and testes, and also to be able to synthesize some steroid hormones. So in addition to the liver, our liver is going to synthesize lipoproteins to help transport cholesterol and fats and other substances in the blood. It's also going to synthesize cholesterol from acetyl-CoA, and we can